you kind of explore your data in a more traditional way, and if your set of predictors are small, you can do that, right? And if you know your samples are good and you don't have problems with sampling issues, your sample size is good enough. Like sometimes people come and say, oh, I have like 50 patients, can you do something? No. Can you fit a regression model? No, absolutely not. It's just your sample doesn't represent anything, right? Unless you are looking for some sort of other studies. Like if you're like a rare drug that is only described to 10 patients in a year or something like that, it has to be super rare.
Welcome to Data Science Dialogues, where industry titans unveil the transformative power of data, exploring cutting-edge techniques and go beyond the limits as we delve into the world of data science. Today we had Farnoosh Sheikhi. Farnoosh is an experienced senior data scientist and biostatistician with a strong background in the healthcare sector. Farnoosh has showcased expertise in data analysis and predictive modeling. Her work history includes roles in various institutions where she has contributed to projects involving clinical data, EMR data, and large-scale data mining. Her commitment to advanced healthcare through data-driven insights is evident in her history of involvement in medical research and data analysis. This video has been powered by Data Science Pathshala. We at Data Science Pathshala are coming up with free mentorship programs. So if you are looking for any help regarding data science, enroll in this program, which includes mock interview, resume preparation, customized paths to upskill in data science, and many more. Link of the registration is in description. Let's dive into it. Manoj, welcome to the pod. I'm truly excited to have you on the pod
Thank
today.
you.
How's it going for you?
Good. Yeah, same, likewise.
So, Farnoosh, I have many friends, who studied biology, even pharmaceutical studies, and I've been helping them, study statistics at times. I believe there are viewers of this pod who would like to more understand what is the role of a biostatistician.
Okay. So, biostat is a wide range. So depending on the field you're working, you would want to have a good understanding of basic statistics. You want to have some sort of programming skills, either knowing like R, which is a, very common statistical software, or SAS. I've seen recently, like a lot of people do Python. But with biostat, the biostat itself comes with clinical applications. So you either get yourself involved with clinical trials. Sometimes you get involved with just like population health type of studies that they are they are interested in some sort of outcome measures. It could be like I've been in healthcare for the past 10 plus years, so everything I've done has been focused on outcomes that are related to healthcare. The other part that you usually get involved with in biostat is a study design. I often get pulled into multiple meetings and team like cross-functional teams to kind of understand what their biostat needs are, what are those areas that they need to identify prior going to a data collection and designing. So it has different variety of functionality that you could get pulled into. I would tell you this, that in order to understand and kind of have a clear vision of like what the teams or the audience want, you want to have a solid statistical background. You want to understand some of the metrics. You want to understand like what are those variations that usually comes in for specific designs, and know the data, like know the challenges that comes with data, like data manipulation, data cleaning, and putting things together. I had a lot of especially in clinical trials, because and like drug developments, there are like a lot of very like experienced scientists that they know how to put things together for drug development, or like maybe like develop a COVID a test, or develop a specific assays that they needs to be for a specific treatment. The part that they don't maybe they don't have a good understanding is that how to put their data together to make it easier for biostatistician to look at it and understand like what are those source of variation and analyze it. So the teams need to come together to kind of come up with solutions for whatever reason or whatever sources that they need, basically analysis or the study to be.
Got it. Now I guess I relate a bit more now.
Mhm.
As we, all know that, artificial intelligence, machine learning, have been transforming industries. and medical industry is not an exception, right? So I remember reading case studies where one uses advanced statistical methods to analyze patient data, or let's say identify potential risk factors, let's say for a rare disease. Can you tell us more about the critical role of biostatistician in such research?
Yes, absolutely. I would tell you this, that the whole, I think, machine learning is part of the statistical analysis. It's engineered based on a specific algorithm to make to kind of unlocking the trends easier. I think the AI has kind of similar, approach, because AI, what is AI? AI is data science by itself, right? So it's what you just train them the same model. You just fit the data and you train it over and over and over and over to get output. So with machine learning, and biostat, they're kind of like they're kind of parallel. They do similar things. Some of the benefits of machine learning is that it has a lot of capability for let's say you have like a large volume of predictors, and you have a specific outcome. Like let's say in healthcare, your outcome could be specific disease, like a rare disease, like if you have some sort of like a genotype disease, or some sort of like a very rare diagnosis that is not common. So with basically with the traditional statistical model, you create that relationship between X and Y being your outcomes, X being your predictors or independent variables you have. Machine learning, it's the same thing, but it has the capability of unlocking the trends better, because the algorithms are utilized, they're built in a way that you can do a lot of cool techniques within that algorithm you choose. Like there has like specifically for like rare disease, because your outcomes are it's like usually 5% of the whole population. Let's say you have 100 samples of patients, or like individuals, like 20 of them or only 10 of them have the disease. It's a very small percentage, right? Like 1% or 2%. So with machine learning, you have the capability of doing a lot of, resampling techniques, or create some sort of like, algorithm to kind of, understand the patterns better. The traditional statistics does the same thing, but it doesn't have all the, built-in functionality that you can utilize.
How do you analyze patient data and identify potential risks for a rare disease, let's say, using ML and AI? Yeah.
So so basically, if you have your your cohort set, so who is in the cohort, like basically find the population of your interest. So let's say for a specific, I can give you like a real example. So one of the studies that I published a paper many years ago. It was around like a kind of like a rare disease in children, and to kind of get a unbiased sample from the population, we just go sample from the database, like a national database that was available to us. And you kind of because and then the disease was for children, like let's say less than 12 years old. So you basically sample similar individuals. So you sample from a population with like, let's say less than 15 years old. You gotta have all their, like, specific not biomarkers, like specific characteristics available to you. So you either, get some of the demographic that you think it could affect the outcome. And let's say if there are information on like the type of blood test, like if they draw any blood, or like if they are any like information on specific disease area, or their history of like their diagnosis with like if their mom and dad knew what's happening, like some sort of like information available to you. And then since once you have that cohort, you can understand, you can kind of explore to see how the disease is related to specific characteristics. Oftentimes, I mean, if your database has all the information, you get some sort of like a basic demographic usually, and there's some sort of like economic characteristic, like if you're living in this area or not. But depending on the design and depending on the available data, once you have your set of predictors and your X metrics, and then you have your outcome, then you can study the relationship. The relationship is the way that I think it's the most successful way to create a model, to predict a model, or even just conduct a very true analysis, is that you got to do some like back end cleaning. You got to create some sort of visualizations to understand how the relationships are working, and think about the outcome. Like if this is an outcome that needs to be a binary outcome, or if it needs to be a categorical outcome, or if it's numeric, some sort of like on having a good understanding of like this is a yes or no questions, or if this is more of like how many times you had this disease, or how many times you incurred this problem, understanding of like what the outcome is. And everything is usually an algebra formula, right? So you have the y equals to like a plus b times x plus c. As so it's an algebra formula. So you take that formula, or either you put it into a regression model, either you do some sort of like traditional stat, you just do let's say you just do some sort of like logistic regression if it's a binary outcome. But if you want to do machine learning, you create some sort of like a assemble tree, like a random forest algorithm, where you basically let's say you have like 30 different predictors, and you want to select the top 10 out of the 30. So you use some of those machine learning algorithms to help you understand what the relationships are look like. And then you take those relationships out of your experimentary analysis, and you put it into the model. Usually with machine learning, there are problems with overfitting, underfitting. There are problems with like how are you accounting for accuracy, and like what are those metrics to think about. So you have to kind of know what you're doing, because the algorithms give you a lot of flexibility around like, do you want to resample from your model, or do you want to just keep the 10, or you want to keep the 15. So you have to have some sort of metrics to help you to kind of optimize the model, versus traditional statistics uh, doesn't give you that much flexibility. You would just like you fit your regression model, and you say that, okay, so I got the model, now I'm going to see if, let's say if model is predicting well, or if it's the model really look like clinically, is it making sense, like clinically, if I see, so let's say for like for something like diabetes, so your model should show that like the high level of glucose or high level of A1C is linked to diabetes, like future diabetes. Let's say you have some sort of biomarkers, or you just do a blood test, or basically your year one, so this year I do some blood tests, I put these basically measurements into a model to tell me, am I going to have diabetes in the next 3 years? Or am I going to get diagnosed with heart attack in 2 or 3 years? So you kind of want to have some sort of clinical background to say that, okay, so if I see, higher level of glucose is associated with future diabetes, that's a right, clinical aspect of it. So the model makes sense. So what I'm saying is that the statistics, the machine learning, all the mathematical formula gives you output, gives you all the relationship, but at the same time, everything needs to make sense, especially in healthcare. Everything needs to make sense, right? Everything needs to be clinically meaningful. It's not just statistics. In my career, I've seen that when you basically combine the traditional statistics with machine learning together, you get the best performing models. That's where, so you don't just want to be, I think this is my own experience and what I've seen over the years, that you just don't want to let the machine learning do everything for you, and without you interfering or understanding the result, right? Because oftentimes you see something, I've seen that like even in some of the papers, you see something and it's like, oh, this doesn't look right. Like, what is like, what are like the relationship in the, wrong direction? This should, based on, the literature, or based on what we, we know, like as humans, like these two are correlated and they should show the relationship. But sometimes machine learning doesn't show it, or maybe show it differently. So having a good understanding of like what the clinical applications are, and with the help of statistics and machine learning, that could be accomplished.
Got it. So a good combination of traditional statistics combined with machine learning and a good iterative process to make a meaningful outcome. Got
Yes,
it.
yes. I mean, with machine learning, it's a parallel, process, right? So you have your machine learning doing things on the side. You do some other, explanatory statistics, see both, direction, and say, okay, these are okay. So I should get some of these, combination. I see these like, relationships. So now I put it into machine learning. So one of the smartest thing, or one of the things I've seen that's like a smart way of doing analysis is this thing. So you do your traditional explanatory analysis. What I mean by that, you do some bivariate, calculation. You look at the relationship between your X and Y, and then you write up, okay, so these are, A, B, C, D, E, F, G, combinations that I got from, my explanatory analysis with traditional way of like looking at X and Y. And then you take that and you put it into machine learning, and you let the machine learning to resample it for you, do all the, repeat that, like a simulation way of like repeat things for 100 times and give me the best output, right? So that's like a good way of like, as a, as a system thinker, because humans are system thinkers, right? Like as a system thinker, you look at things and you're like, okay, now this is like, showing some trend. Now I take that piece of information and put it into AI, or put it into, machine learning. So that way you can have a very, more, effective result, a model that could be, used like some of the healthcare maybe utilization I've seen. The other thing that, one of the other advantage of, I think machine learning is that if you have some sort of huge database, like I've done work for like some of the hospitals that they have, 400 labs, and then they have 400 different procedures, and they have like 200 different, diagnoses. And they just want to, let's say the outcome is just to flag patients with high risk. So they just want to flag in their system, they just want to flag things. They say that, are you a high risk? Yes versus not, right? So for those type of questions where you're not looking at the specific diagnosis, or your outcome is more generalized, like like you could have a diabetes, you're high risk. You could have like the other person has like, kidney disease, that's high risk. It's more, it's more generalized than like a specific disease or rare disease. For those, that's where machine learning is super useful, right? So you put your, let's say 1000 matrix of columns into machine learning and say, find me the most, like, let's say the high cost combination of things, and then flag those conditions for me or something like that. So for those things, you can use machine learning because you have a black box of things and you don't know what you're doing. Like, you have to find things in it. So that's useful for, but if you have something, if you have a specific disease or a rare disease, the best thing to do is that you kind of explore your data in a more traditional way. And if your set of predictors are small, you can do that, right? And if you know your samples are good and you don't have problems with like, sampling issues, and your sample size is good enough. Like sometimes people come and say, oh, I have like 50 patients. Can you do something? No. Can you fit a regression model? No, absolutely not. Because it's just your sample doesn't, represent anything, right? Unless you are, looking for some sort of other studies. Like if you're like a rare drug that is only described to like, 10 patients in a year or something like that. It has to be super, super rare. But if you're like looking for a specific, disease, you want to think about like what are those, prevalence and incidence rate that like maybe you can find it from CDC website and say that, okay, so these prevalence are around 5 to 10%. And there are studies that they conducted it for, like 100 to 200 patients. So you want to stay in that range of sample size to get meaningful results. So sample size is another, issue with rare disease.
So, Farnoosh, talking about sample sizes, as I, deal with data science cases, one of the biggest challenges that I face is, giving the stakeholders the idea of what data is required to make this particular model or to solve this data science case. So, are there any similar challenges that you face as a biostatistician?
Yes, absolutely. I think sample size estimation is one of the main things that we always deal with, especially for clinical trials, especially some of the smaller studies that like if we want to like, develop like some sort of a diagnostic, test, we have to think about that. Um, so here's the little hint or thing that I say. Think about your sample size with the effect size you're thinking. These two are always together. So if you have some sort of, studies that you say that, okay, so for this study, I'm going to get that number of positive samples. So let's say you're looking at, a test that your results are going to be either positive or negative. In the population that you're thinking, you think that you're going to get a lot of negative because people don't have that specific disease. So they're mostly like negative, results for that disease. So you want to have a lot of positives to kind of understand what the, prevalence is. So either you have to assume some sort of, assumptions around like, okay, what are those, like similar disease or similar, studies that have been done? What are those rates? Kind of like understand like what are the common rates, and then do the sample size calculation depending on like if you're looking at the, changes over time, or if you're looking at two different, group, that one group has the disease, one group doesn't have the disease. How do you do the calculation? So the effect size is the main thing to think about when it comes to sample size calculation. If you are doing modeling and like cohort studies or doing a lot of machine learning, depending on the number of predictors you have, I think there's a couple of papers that they say you don't want to have, for example, you don't want to have a data with like, let's say 100 observations and 200 predictors. You know what I mean? I think it's a square root of observation.
So you don't want to have a lot of variables as compared to the number of observations, and at the same time, you don't want to have too many observations as compared to the number of
Yes,
variables. So there is some, relation between these two in order to have a, decent model to be framed. Both the cases have their own issues. Yeah.
exactly.
So I'm not aware if there is a formula for that, but I'll figure it out for sure.
Yeah, I think it's a square root of observation. I know for some of the, have to go pull it out of my, my previous work. I think there's even in random forest, which is a machine learning algorithm, and it's been studied to be one of the best algorithm to do predictive models, and you get good predictions out of the random forest algorithm. There's a formula that says if you have if you have that many predictors, use this formula to kind of like adjust for the number of predictors that your algorithm select. Like you have some sort of like, you can choose, because there's overfitting and underfitting, right? With the machine learning, you want to choose right amount of predictors to include into your model so your model doesn't overfit. Like if you have too many predictors, the model is overfitting, right? So you want to have that ratio. But there are documentations around that for sure.
So, Farnoosh, I'm really curious about how your daily routine as a biostatistician looks like, and more importantly, what is the best part you believe is being a biostatistician.
Oh, my days. So most of my days are around meetings, a lot of meetings to kind of understand what the team needs. I try to block some time for myself to do, to run codes, do some reading, writing, whatever I need to do. But it's a combination of meeting with different teams to understand what they're doing and what are their needs are for the analysis. And then the other portion of my work is just like working with data. I do a lot of programming every single day. I use R and R Studio mostly because it's open source, and I've been using R for the past 10 years. And the other part that I need to make sure that I handle it well is that the communication of the results. So oftentimes uh, I have a lot of like scientists that they are expert in their fields, but they don't know statistics. So I write up the results for them in a way that they understand it. It's like simple. You explain, like, let's say what the odd ratios are, or like how they can how they can utilize that results into their findings. So some of it it's writing. So it's a combination. If I give you some percentages, it's like there's like 30% of my time that is usually around meetings. I'm working with other teams to understand what they need. The other, 40% is me analyzing the data, looking through some of the results. And there's like, the other 10-20% are communication, writing up things for the team to understand. And I also use some of my time to see what's out there, if I need to kind of do some learning on my own, or if I need, I go to like this like different blogs every day to see what people are doing, what are the new packages they use. So some learning, like every week, like I try to do some learning as well.
Got it. So I would say for me, it is, drawing the inferences out of the model or the statistical process that you go. That's the best part
Oh, the
for
best
me as a data
part.
scientist.
I think the best part for me is the programming. I enjoy I enjoy data visualizations a lot, and I go like I go crazy. Usually like generating too many things. Like I had this like project that I was doing on that I published a paper recently, and I'm the first author on it. I spent 60 slides just looking at different type of plots, and like some of the team members were like, oh my god, this is like too many. But like for me it was just like fun to see how things are moving together, like the trends. Like if you look at female versus male, or if you look at female within like age of like less than 45 and then above 45, like the trends of like the data that's like moving together. So the data visualizations, I think it's my favorite part of the work.
Yeah, yeah, I understand that it's one of the most important
Yeah.
parts, I would say, because stakeholders may not understand the statistical terms or values, but they will surely understand the visualizations per se. Yeah.
Yeah,
So
that's fun, right? It's like doing,
yeah, that's
yeah,
fun.
it's like doing art, not just numbers.
Amidst the initial chaos, during the COVID-19 pandemic in the recent past, I, recall hearing stories of, heroics of the frontline healthcare workers. Could you, share a personal experience of, how you as a biostatistician collaborated, with medical professionals at that time, despite the uncertainty?
Oh, during COVID. So Quest was developing a lot of, a lot of diagnostics and tests. And we were part of a team. There's actually, we published a paper. If you search Quest COVID, I think the paper will come up. Uh, we published a paper, and one of the main things for our team or biostat to basically be involved and capture was that the precision, the accuracy of the test. So there were a lot of trials or pilot studies that we had to make sure that how to basically estimate the accuracy of the test, how to look for the precision of the test, like if it's real positive or negative. There was there were some very involved statistics that was conducted to capture that. And prior to that, we did like as I said, like power calculation for sample sizes to make sure that the studies that we are looking at, there is enough, we have enough power for the effect size that we were thinking. So the precision and accuracy was one of the main things that we observed. But overall, I think throughout COVID, we had, I think the work volume was super high. So Quest got a lot of help with like other, like they were like many things that were happening on the operation side that I wasn't involved with it. So I wouldn't know that. But as far as the diagnostics and, the statistics, like looking at the precision and looking at, the accuracy of the test, it's like what those were the biostat, functionality during that time.
Got it. So, before we continue, a big shout out, and appreciation for all the,
Yes.
frontline workers and medical professionals who demonstrated, incredible resilience during those testing times, right? Considering the possibility of future pandemics, okay, have you been involved, or if biostatisticians are involved in initiatives, that enhance the preparedness and response through their work as a biostatistician?
Yeah, for it's not just pandemic, like for any, I think all the diagnostic or all the pharma companies, they are constantly developing, right? They're developing either a new drug or they're developing a new assay or a new test. There's a new genetic test that I think Quest is gonna launch soon. That's it can similar to you heard of 23andMe, that you do like a test, and it gives you some sort of like history of like your genetic maybe like this, not disorders, or like genetic components that could cause some sort of cancer, or it could be a flag for a specific disease. So I think within healthcare, that's the common practice. Always looking for opportunities to save lives, right? To make a healthier life. So there's always that progress of like, how can we predict, let's say, specific disease? How can we predict, let's say, preterm birth, because it's super expensive and some of the children die like when they are born and they are preterm, right? So there's always something, there's like many areas that there's always development and work in progress to kind of uh, unlock some of the things that we could save lives. So I think that's always part of the healthcare I think industry.
Got it, got it. So, Farnoosh, in your experience, given that biostatistics, I believe, is not a very new field,
Mhm.
but at the same time, not very widely known. But have you mentored or guided an aspiring biostatisticians and witnessed their growth in the field?
Yes, I have. At Stanford, I work at Stanford School of Medicine for over 3 years, and I was part of basically a team that there were interns coming from university, Stanford University, to School of Medicine for research. So I was supervising, 2 or 3 interns every summer for their internship to kind of show them how they can utilize the statistics for their research. oftentimes they wanted it to publish a paper, so they had, some sort of like specific, study that they needed help. We would tell them like I would tell them like what to do exactly, what are those resources they need for statistics. And I had a lot of other cases that the interns really wanted it to learn programming and use R. So I had to, I was like sitting with them to just show them how to load their data and like how to conduct, let's say, t-tests between the, mean of group 1 versus group 2. Yeah, I had that experience a lot at Stanford. Here at Quest, everyone has, oh my God, like over 20 years of experience. Everyone is senior level, so it's a lot less of like, newcomers. It's more of like very, highly educated with many years of experience. But I definitely enjoy working with interns. It's great to see that they even published papers and made a huge progress in their career right after graduation.
Got it, got it. So, for a data scientist, who aspires to pivot into biostatistics, have you ever encountered someone like that who made a successful transition? Or, what can someone do in order to transition to the same?
Like from data science to biostat.
Yes, yes. For example,
In the other way.
no, no. I mean, for example, I am a student. I mean, I studied biology till standard 10. Then I dived into, physics, chemistry, mathematics, and I never had biology after that. So let's say I want to dive into, to become a biostatistician. So what should one do per se?
Oh, if you want to become a biostatistician, yeah, get a degree in statistics. Honestly, there's no other way around. Like, I mean, I'm sure there are other ways around it, but to understand, statistics and clinical, aspect of basically biostat, you want to have some sort of training. I know some of the there are so many online resources these days that you can study for 1 year. Like, let's say at Berkeley, I know there's like online programs, or at MIT, there are online programs that you just want to get exposed to the type of studies that are happening in the biostat field. And in the biostat is, it could be in a clinical field, population health, usually. It could be in drug development, clinical trials, or it could be in medical devices. Like companies like Medtronic, or the companies like not Genentech. Genentech is all diagnostics. There's a BD. It's like the BD company, Medtronic. Um, there's a couple of other. Intuitive Surgical. That's another company that they are developing robots. And they do biostat for the medical devices. So their medical device has to be really precise. So they measure the like precision of their medical devices and different parts. So that's another field. So it's either the medical device companies, the pharma, drug development, or the clinical, developments and like population health. So these three areas that have been encountered in the past 10 years. But overall, to come to biostat, you need to have a, solid understanding of statistics, knowing, what test gets used for what, scenarios. Um, are you supposed to, look at regression, or are you supposed to look at correlation, or are you looking at clustering? Like learning some of the basics. I would say that most people who do data science, they don't know biostat. Like they know a lot of, data manipulation and data visualization, but they don't know a lot of clinical or the biostat field, where it's when it comes to like designing a study for a clinical trial, let's say, or sample size estimation, or some of the other challenges that comes in the field of biostat. Um, so that's why I told you I've seen the other way around. So I've seen a lot of biostat going to data science, but not data science coming to biostat because of that, the level of understanding of like a statistical methodology, because the methodologies are what it's the application of biostat, right? Like, are you using, this method or not that method? And the other challenge is that some of these clinical trials that are need to get approved by FDA, there's like documentation on like CSLI, which is a clinical laboratory, institute, that they publish all these, documentations or guidelines. So you have, and it looks like a textbook. So you have to understand the methodology in those. So there's a lot of methods, a lot of understanding of like the statistical methods versus the data science. It's more of like how to how to use the data, what algorithm to use. So it's less, I think, methodology.
Got it. Got it. Got it. So, lastly, you worked closely with, medical experts and, navigating and learning and understanding medical aspects relevant to your research. What is the most, challenging part that you find being a biostatistician?
Oh, that's a great question. I think the most challenging part is explaining the statistics or the methodology to non-stat audience. That's the main part. That's hard. That's hard thing to do. And you can accomplish it easily because it's a hard concept, right? Like some of the concept, or some of the science behind it is like pure mathematics. So it's hard to kind of communicate the mathematics to someone who has like zero, is like studied biology for like many years, right? So it's hard to like kind of say that, oh, this was like based on this formula. So what I've done in the past, and like any time that I felt like I can't, like I don't know what to do, like how to explain this is hard, I would go to publications and see the type of method that you use and the type of summary they provide, because the language publications are easy to to explain, and you can kind of use the same type of language for someone who doesn't know enough about statistics and math. But that's been always a challenge. Like, it's not something I had a professor at Stanford that he asked me what the variance is, like mean variance, standard deviation. I'm like, what? Like variance? Like everyone should know variance. Like, and I'm like, okay, this is the distance. Like if you think about like series of data points and you get the averages, so think about the distance from the average to each data point, and the variance, like standardized that distance. Like thinking about like how to think about like, and the p-value, like what is p-value, or like how do you, like, what is the p-value 0.05 different from 0.01 or less than 0.001? Does that mean that my p-value is more, does that mean my effect is stronger if my p-value is smaller? I'm like, no, it's the sample size. The smaller the sample size, the p-value will affect, by the sample size, right? um it's been a challenge. It's always a challenge. So it's kind of like, kind of know what the person knows, and like how to bridge the gap between the two. You learn as as you go. Yeah, yeah,
Yeah, yeah, I absolutely relate. I absolutely relate. Uh, talking about the p-value, it took me myself being studying statistics for so many years. It took me more than I expected to comprehend what p-value is and how to determine the level of significance and so on. So yeah, I mean, and then comes describing it to the person who doesn't know statistics. Yeah, I mean, or has doesn't comprehend statistics that well. So Faroosh, thank you so
yeah.
much for
Of
joining us
course.
today. and sharing your personal insights and stories. I guess I have a much better and clearer idea about biostatistics now. So yeah, thank you so much
Yeah,
for
of
making the
course.
time.
Good luck, and hope you enjoy whatever, whenever wherever you go, enjoy it, right? Because it'd be
Yeah,
full-time job, 40, 40 plus hours a week, so you want to enjoy yourself.
very true.
Right?
Can't agree more.
So that's
Yeah.
important. Like, I mean, this is kind of like a personal note. But I studied physics on my undergrad, and my
Okay.
master degree, I got a master degree in statistics. And the reason I got to statistics was that I always enjoy mathematics. And I had like some
Mm-hmm.
of the challenges of like go do engineering or do like computer science, but the mathematics always made me feel good and made me happy and like solving problems. So that like personal, interest is always the key to any career.